During interphase, many cells make proteins. They make very specific proteins depending upon the type of cell. Protein synthesis begins in the nucleus. Most specifically, it begins in a gene. The gene carries the information to make a specific protein. This information is written on to an RNA. The RNA is taken out of the nucleus and into the cytoplasm, where it will code for the formation of a protein. So we start with the gene. Here's the gene in purple, the double helix, which is the DNA. A small part of it is the gene. This gene carries the recipe to make the protein. And this recipe is written on an mRNA molecule. It's called a messenger RNA molecule, which is taken to the cytoplasm. And this recipe is used to make a protein. Proteins are made up of amino acids, and this recipe has all the information necessary to string up these amino acids together to make a protein. The first part of the reaction from DNA to mRNA is called transcription. The second part of this process from mRNA to protein is called translation. DNA to RNA to protein. That's the movement of information or the flow of information. Transcription takes place in the nucleus and translation takes place in the cytoplasm. This is the overview of protein synthesis. Imagine that you are in a library carrying very old recipes to make a cake, and you want to copy out the recipe. You don't have your cell phone with you, just a notebook and a pen. So you're going to rewrite that recipe in your own handwriting in your notebook. This is what we call transcription, taking a piece of information from somewhere and rewriting it in your own handwriting, for example. So that's what happens in protein synthesis. During the first stage of protein synthesis, DNA carries the recipe, which is the gene. It cannot leave the nucleus, so the information is written on to an RNA molecule. We call this RNA molecule a messenger RNA molecule, or mRNA. The enzyme that makes this possible is called RNA polymerase, and it helps to add one nucleotide at a time to the growing mRNA chain. Remember, this whole process takes place in the nucleus. You want to imagine the nucleus as a library for all the recipes to make proteins. We only need one, and so we carry it out with the mRNA or the messenger RNA that carries the message or the recipe from the nucleus to the cytoplasm. And we use the word transcription because we're using the same language, which is the nucleotide language, where we use letters such as ATGC or AUGC. In the RNA, whenever an A is encountered, a U is used. RNA does not have thymine. Instead, it has uracil. So A goes with U and G goes with C to make the mRNA molecule.